Hello students, how are you all? Welcome or welcome back to my space. Hope you all are doing and learning well. If you like my videos, then don't forget to subscribe my channel and you can share to your friends as well. So let's talk about today's video. It's all about the cell organelle lysosome. I'm dividing this video in two parts as this topic is too large to be completed in single video. So today we will learn about the structure of lysosomes, how lysosomes are formed and little bit about how they work. In next video we will look at the functions of lysosomes. So without further ado, let's get started. Introduction. Lysosomes. What are the lysosomes and what is the etiology? So, lysosome is taken from the Latin word combining two words. The first one is lyso and the other is soma. The lyso means to loosen or to breaking up and soma means body. So, these are the body that lyses, or these are the structures that are used to break down the complex material into smaller or simpler ones. So, it is a membrane-bound organelle found in all eukaryotic cell or animal cell. Suppose, just look at this picture. This is our typical eukaryotic cell, okay, which contains so many organelles. These are so many organelles. Let me use this, okay. These are so many organelles that are present inside the cell. This is the nucleus, the endoplasmic reticulum, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, centrioles, and these bodies that are yellow in color are lysosomes. So these yellow colored spherical bodies are known as lysosomes. They are spherical vesicles containing many hydrolytic enzymes which assist in lysing of biomolecules. So these spherical structures that are present inside an eukaryotic cell are used for lysing the biomolecules. These are used for breaking down the biomolecules that may be proteins, lipids, carbohydrates, etc. So these basically break down the complex into simpler ones. So if we magnify this particular spherical structure, we can see the structure of lysosome. So the lysosome is having spherical sacs which is enveloped by a membrane which is quite similar to our plasma membrane. Hence, if we magnify this particular layer, we can see that it is composed of phospholipid bilayer. Okay. It is composed of phospholipid bilayer. And inside this, you can see there are so many structures. Some are square, rectangle. Actually, they are representing the hydrolytic enzymes. Almost more than 50 types of hydrolytic enzymes are present in this particular vesicle, or we can say sac. So these structures are hydrolytic enzymes. Christian D. Dewey, a Belgian biologist, discovered it and coined the term lysosomes. He received a Nobel Prize in Medicine or Physiology in 1974. So the Christian D. Dewey is a Belgian biologist that discovered the lysosomes, and he also received Nobel Prize in Medicine or Physiology in 1974. So this all about the background or the history behind the lysosomes. Lysosomes are thought to be a trash can of a cell, or we can say garbage disposal of a cell, as it digests the foreign stuffs, older or dead organelles that are non-functional or under stress. So, lysosome is represented as the organelle having diverse functions. They are also known as the cleaners of the cells waste, as they digest the waste with the help of the enzymes that contain. So we can say lysosomes work as a trash can of a cell, or they are the cleaners of a cell. Now the structure. Lysosomes are membrane-bound organelles, as I just told you, or the spheres which are made up of lipid bilayer that encloses a fluid called lumen containing variety of hydrolytic enzymes. So inside a cell, if this some structures like spherical vesicles that are present inside which they contain some hydrolytic enzymes, where they contain hydrolytic enzymes. This portion is known as the lumen of the sac. This is the lumen, and which contain so many hydrolytic enzymes. Hydrolytic enzymes. So you can see in this picture, this is a cross section of a lysosome. You can see this is having an spherical structure which contain hydrolytic enzyme inside the lumen. This is the lumen, and these structures that you are seeing, these are known as hydrolytic enzymes. So I am telling again and again the hydrolytic enzymes. Let me tell you what are these. We know that all the enzymes are proteins. Okay, we all know that all enzymes, we can say all enzymes are proteins, but all proteins are not enzymes. So these, the lumen containing the enzymes, are initially a protein, or we can say these are proteins. So hydrolytic enzyme means those enzymes that use water to break chemical bond, result in during or breaking a larger molecule into smaller molecules, or in other words we can say that they digest the molecules so that it can easily use up by a cell. Various enzymes are the enzymes that are present inside this lumen are lipases, which break down lipids, phosphatases, glycosidases, peptidases, which break large protein molecules, and nucleases, which digest nucleic acids. So generally they are very small, ranging from 0.1 to 0.5 micrometer. Sometimes they can reach up to the 1.2 micrometer in diameter. The pH of the lumen is 4.5 to 5, which is optimal for the activity of the hydrolytic enzyme. So if we talk about the hydrolytic enzymes, they are functional in particular pH. So the pH in which the hydrolytic enzymes works are should be acidic in nature. So the pH of the lumen is 4.5 to 5, which is slightly acidic and optimal for the function of these hydrolytic enzymes. Besides degradation, they are involved in plasma membrane repair, apoptosis, secretion, and autophagy, etc. So, the main function of the lysosome is to break down the complex molecule into simpler ones so that it can use up by the cell. And beside this, there are so many other functions, like they are also helps in plasma membrane repair. It helps in apoptosis, secretion, and also in autophagy. Now, why lysosomes are known as suicidal bats? Lysosomes acts as the waste disposal body of a cell by digesting undesirable stuff both from the exterior as well as the inside of the cell. We know that we had just seen that the lysosomes acts as a waste disposal system or waste disposal body which digests the undesirable, that is not is going to any use of a cell. It may be from inside the cell or it come from outside of the cell, like any pathogens or foodstuffs. So it basically helps in digesting the undesirable stuff that is no more of any use of a cell. But sometimes cell is in stress condition or damaged or infected. Okay, so the lysosomes get burst open by destruction of its cell membrane and all the hydrolytic enzymes get released, which digest their own cell, causing cell to die. This process is known as autolysis. Auto means self and lysis means disintegration. So it is also happen when the cell is not getting proper nourishment or nutrients. It autolyses itself and become food for other neighboring cell. So let me make you understand with some diagram. Suppose this is these are the cells. Okay, these are the cell. This one cell that is at the center is in stress condition. This cell is stress because it's not getting proper nourishment or nutrients. So what it does, the lysosomes of this cell, the lysosomes of this cell get burst open and pour all its hydrolytic enzymes to the cell, which causes the cell to die. So this cell is going to die and disintegrated into very simpler components. We all know that a cell is made up of lipids, proteins, nucleases. So the simplest form get used by the neighboring cells, and they use this cell as its nutrients. So let's take a look once again. The hydrolytic enzymes may end up damaging the lysosomes themselves. Okay, if the cell is in stress condition, or we can say if the cell is is getting infected, so the lysosomes itself burst out and release all its hydrolytic enzymes, which ultimately leads to cell death. This phenomena is known as autolysis. This process is very important and you have to learn it to understand it very well. Okay, So this phenomena is known as autolysis, where auto means self and lysis means disintegration. So autolysis, we can say autolysis, the self-killing of a cell with the help of lysosomal hydrolytic enzymes. Next. Now we know that such a savior is these organelle for a cell. So let's talk about how they are formed, how lysosomes are formed. We know that lysosomes are vesicles budding off from the Golgi apparatus. But these vesicles contain hydrolytic enzyme. So next question is where these hydrolytic enzymes comes from. We also know that all the enzymes are proteins. We know that all the enzymes are proteins. Okay, so let's go back and take a look how proteins are formed. We know that suppose this is our nucleus of a cell. This is nucleus. Okay, and inside which we have our DNA. This is DNA. We know that the process by which mRNA is formed from the DNA is known as transcriptase. So from this DNA, this is DNA. From this DNA, mRNA is synthesized. This is mRNA. So from the DNA, the mRNA is synthesized in the nucleus, and this process is known as transcription. This mRNA comes out through the nuclear pore. Now this mRNA, it gets out from the nucleus through the nuclear pore complexes. So this is mRNA. Wait, let me make endoplasmic reticulum also. This is rough endoplasmic reticulum. Okay. In the cytoplasm, proteins synthesize with the help of who synthesized proteins in the cytoplasm. These are the protein synthesizing bodies known as ribosomes. So ribosomes are help in synthesizing proteins. This is the ribosome, this is mRNA, and these ribosomes are involved in synthesizing long peptide chain, or we can say proteins. The ribosomes, we have learned about that the ribosomes are stuck on the endoplasmic reticulum. Therefore, these endoplasmic reticulum that are near to the nucleus looks rough in appearance. Thereby, these are known as rough endoplasmic reticulum. These ribosomes stuck on rough endoplasmic reticulum, and further proteins are sucked up in the lumen of RER. So, this particular ribosome, which are synthesizing the nascent protein are sucked up inside the lumen of RER. So this protein get inside the lumen of RER, where proteins get folded up, or we can say modification of proteins takes place. Okay, inside the rough endoplasmic reticulum, the folding up of this nascent proteins takes place. This folding up is necessary because it makes the protein functional. Inside RER, the most important thing is happened. What is that? This is the attachment of a Mannose group. So let me take this blue color. Inside the lumen of RER, a Mannose group is attached to this protein. This is the Mannose. This is the Mannose. Okay. Let me draw this again. Suppose this is our protein, and we know that protein has two terminus. The first one is amino terminus and H3 amino terminus or N terminus, and the other one is carboxy terminus. Okay, so this Mannose group is attached at the N terminus end of a polypeptide chain. This is Mannose. This is Mannose, okay? So the process by which this Mannose group is attached at the N terminus end of a peptide chain is known as N-linked glycosylation, and this step is very important in the processing of lysosomal proteins or lysosomal enzymes. Now from the RER, this is budded off. Wait. This protein which is tagged with Mannose is budded off, containing peptide with added Mannose. These vesicles in the form of vesicles. So the RER membrane is pinched off, which contain the Mannose added peptide. Okay. In the form of vesicle. This is the vesicle. Now one more important, how this vesicle knows that it has to go to the Golgi apparatus. So in this we have to know one more thing, that this vesicle, this transported vesicle is coated with special type of proteins known as COP2. So this vesicle is having the COP2 type protein, which destined this particular vesicle into the Golgi apparatus. Okay, so let me draw the cisterns of Golgi. Now this vesicle get attached to the cisterns of Golgi apparatus. Now inside the lumen of the Golgi apparatus, there is another main important process, that is phosphorylation of this protein with mannose takes place. Now inside this, a phosphate group is going to add to this mannose protein. So now we have mannose 6-phosphate protein. This complete complex is known as mannose 6-phosphate. Okay. So inside the lumen of the Golgi apparatus, a phosphate group is added to the carbon 6 of mannose. Now, what is the next? This specific now, when it budded off through the transphase of a Golgi apparatus, this particular is pinched off, okay, in the form of a separate vesicle that is ultimately known as our lysosome. Okay, so this is the lysosome that contain the mannose 6-phosphate. This specific lysosomal tag, that mannose 6-phosphate, allows proper packaging into vesicles which ultimately destined to the lysosomal system. We will learn later what is the lysosomal system. Lysosomal system. So let's take a quick recap that how lysosomes are formed. As I just told you, lysosomes are protein gene transcribed in nucleus which is controlled by the transcription factor EB, TFEB. So inside the nucleus we have the transcription factor EB which used in the formation of mRNA. Okay, now mRNA transcript, this mRNA transcript is exit the nucleus through the nuclear pore complex into the cytosol, okay, into the cytosol where they are translated by the ribosome. So these are translated by the ribosomes. This is the ribosomes and this is going to synthesize the polypeptide chain from RNA. Now the nascent peptide chain translocated into ARIA. Now this is stuck up inside the lumen of RER where they get modified and a mannose group attached to it, and this process is known as N-linked glycosylation. Now the next is the liposomal proteins is exit through the endoplasmic reticulum via vesicle which is coated with the COP2 proteins. These are the coating proteins type 2. Now these COP2 vesicles direct lysosomes proteins or enzymes to the Golgi apparatus, where addition of phosphate group on the liposomal proteins takes place, and this process is known as phosphorylation. This specific lysosomal tag, that is mannose 6-phosphate, allow proper packaging into vesicles, okay, and this vesicle is going to perform its duty. Next. Now, how lysosomes work. For understanding the process of how lysosomes works, we have to first understand about the process of endocytosis. Okay, so it is a process by a cell which engulf any stuff which may be bacteria, virus or food particle, or we can say it is a cellular process in which stuff that to be digested or break up is surrounded by an area of cell membrane which bud off inside the cell to form vesicle. Then this one is known as endosome. So let me explain you with diagram. Suppose this is our cell. Okay, and here is a foreign particle. Suppose this is a foreign particle. It may be bacteria or virus. So this is the stuff that is going to be digested. Now what will happen? This cell membrane is going to invaginated this particular stuff inside the cell, like amoeba or macrophages does. So what will happen? And ultimately it bud off inside the cell. Okay. So now this one is known as endosome, and this process of engulfing any foreign particle is known as endocytosis. This particular vesicle is known as endosome. Okay. Now there is one speciality of this endosome, that the outer membrane, that is plasma membrane, is having lots of proton pump. It's having lots of proton pump. Okay. This is our primary endosome. It's having lots of proton pump that are used to pump inside so many protons from the cytosol of a cell. So many protons are get inside this endosome, which ultimately make this endosome more acidic. Okay, this endosome, which are having so many proton pumps, made them acidic in nature. So inside this endosome, the environment is more acidic as compared to the cytosol. So this one is the late endosome. Okay, this one is known as late endosome. This is late endosome because it continues to pump protons inside the lumen and become more acidic. Now what? This late endosome is now get fused with the lysosome. Okay. So suppose this is our cell and this is our late endosome containing the foreign particle. It gets fused with the lysosome containing hydrolytic enzymes. So this is our lysosome. And this is late endosome. They both get fused together they both get fused together and the hydrolytic enzymes get poured to this foreign stuff and break it into simplest form or in smaller particles that is used by the cell. Okay, so next what? Most of the stuff is used by the cell, and those stuff that is not going to be of any use or that is waste is exported out through or is spit out by a cell via process known as exocytosis. Exocytosis. Okay, so the function of the lysosome starts after the formation of endosome via process endocytosis, as we just talked about. Now, after leaving Golgi, the hydrolytic enzymes filled lysosome fuses with a late endosome, and this late endosome is having very acidic environment inside the lumen, which is about 5.5 pH. This acidic environment causes dissociation of the lysosomal enzymes from the Mannose 6-phosphate receptors. That these enzymes break down or digest the engulfed stuff and spit it out from the cell via process known as exocytosis. That's all for today. And if you have any query, please text me below and share your suggestions about the topic you want me to cover in upcoming videos. Thanks for watching.